Hello everyone. We will continue today talking about fatty acid oxidation in part 2, and this lecture include fatty acid transport from cytosol to mitochondria. Like in the previous lecture we have talked about how fatty acids are transported from dietary lipids to the different cells, cytosol of different cells. Similarly, fatty acids from stored fat in adipocytes that also could be activated because of presence of glucagon, and from adipocytes fatty acid transported to the cell for oxidation purpose. In today's lecture we'll talk how fatty acids from cytosol they are transported to mitochondrial matrix, because beta oxidation occurs in mitochondrial matrix. All the reactions of beta oxidation. Then we'll take one example of palmitic acid and we'll calculate total ATP produced from palmitic acid molecule. Then we also talk about beta oxidation for unsaturated and odd number fatty acids. Beta oxidation also occurs in peroxisomes also along with mitochondria. So what are the differences in beta oxidation reactions when it occurring in peroxisomes and in mitochondria? That also we will talk in this lecture. And finally, beta oxidation in germinating seed. In germinating seed, beta oxidation occurs in glyoxysomes. So all those reactions which occur in glyoxysome, those also we'll talk in today's lecture. The complete oxidation of fatty acid consists of 3 stages. In stage 1, when fatty acids are present in cytosol, that is transported to mitochondrial matrix, and just after entering to the mitochondrial matrix, fatty acid start oxidizing, and the oxidation occurs using this process called beta oxidation, where 2 carbon atoms from fatty acid they are removed in form of acetyl coenzyme A. For example, if fatty acid contain 16 carbon atom, in that case total 8 acetyl coenzyme A will be produced after beta oxidation. And during beta oxidation, electrons, they are also electron carriers, NADH and FADH2, those are also produced during beta oxidation reactions. And these acetyl coenzyme A, which were 8 molecules, those enter to citric acid cycle, and more FADH2 and NADH molecules are produced. That's in stage 2. In stage 1, beta oxidation reaction occurs. In stage 2, acetyl coenzyme may enter into citric acid cycle to give rise to carbon dioxide and more NADH FADH2 molecules. In stage 3 of oxidation, these NADH and FADH2 they enter into respiratory chain, and eventually those electrons from these carriers those are utilized for oxidative phosphorylation of ADP to ATP. So total number of many ATP molecules are produced eventually after electron transport chain. And the site of beta oxidation is mitochondria. Even in case of sugars, polysaccharides and sugar, they enter to cytosol. After entering to cytosol, sugars are converted to glucose, and this glucose is converted to pyruvate. This process, this we have talked already about this is called glycolysis. So glycolysis, after glycolysis this pyruvate formed, this enter into mitochondria, and after entering, pyruvate is converted to acetyl coenzyme A. Similarly, fatty acids they also enter to cytosol first, and in cytosol fatty acid has 2 choices. Either it can go to mitochondrial matrix if oxidation is required, energy is required from oxidation of fatty acid, or this fatty acid can be used for synthetic reactions. It can be incorporated in other molecules to give rise synthesis of other lipids which could be stored. And the transportation of fatty acid to mitochondrial matrix that occurs with the help of specific transporter molecules which we call carnitine fatty acyl carnitine shuttle system, and after entering to mitochondria, this fatty acid go for beta oxidation reactions, and eventually acetyl coenzyme A is formed. This acetyl coenzyme A, this enter into TCA cycle. Large amount of energy generated by oxidation of fatty acid, or there are 4 reactions in beta oxidation, जो कि in every cycle of 4 reactions, the oxidation occurs at the beta position of fatty acid. Beta position means this is COH group, this called alpha, which attached to COH group is alpha carbon, and another next is beta carbon. So oxidation occurs in every cycle, oxidation occurs at the beta position. That's why it is called beta oxidation. And in one cycle, 2 carbon atoms of fatty acid, they are removed. They are removed in form of acetyl coenzyme A. That's what happens in case of beta oxidation. So दो carbon atom fatty acid के one after another, they get removed in form of acetyl coenzyme A in beta oxidation. So beta oxidation में तो every cycle के बाद the fatty acid produced will be 2 carbon less, क्योंकि जो दोनों कार्बन है वो दोनों कार्बन acetyl coenzyme A फॉर्म करके they are released from the fatty acid, and it will keep repeated. cycle तब तक चलता रहेगा जब तक पूरे कार्बन आइटम फैटी एसिड के they are converted into acetyl coenzyme A. And one more thing here, fatty acids with carbon chain more than 14 carbon. We know that fatty acid जो है their chain length varies from say 3 carbon to they go beyond 20 carbon atoms. So अगर 40 carbon से नीचे के fatty acids हैं, they do not require activation. cytosol से direct वो जो है ट्रांसपोर्ट हो जाते हैं into mitochondrial matrix. लेकिन जो 14 कार्बन से लेंथियर हैं, ज़्यादा लंबे फैटी एसिड्स हैं, उनके लिए दे रिक्वायर एक्टिवेशन स्टेप. एंड दे ऑल्सो इफ दे आर लेंथियर कार्बन, दे आर मोर नंबर ऑफ कार्बन कंटेनिंग लॉन्गर फैटी एसिड्स, एक्टिवेशन के साथ उनका ट्रांसपोर्टेशन भी रिक्वायर्ड है. दे कैन नॉट ट्रेवल अक्रॉस द मेम्ब्रेन ऑफ माइटोकॉन्ड्रिया बाय डिफ्यूजन. they are facilitated by a system called carnitine transporter system. Carnitine shuttle भी उसको बोलते हैं. So that's for longer fatty acids. So in the first step, fatty acids, they are activated. And how do they get activated? By the action of enzyme called acyl coenzyme synthetase. Acyl coenzyme synthetase transfer coenzyme from coenzyme A to the fatty acid, and fatty acid is converted into fatty acyl coenzyme A. Same time it use one ATP molecule. ATP के in fact 2 phosphate groups are hydrolyzed here, 2 phosphate bonds are hydrolyzed. So that's why ATP is converted to AMP here, not in ADP but AMP. And there are 3 isoforms of this enzyme. Each isoform act on a specific length of fatty acid. एक आइसोफॉर्म है जो शॉर्ट, अगर 12 से छोटे इनफैक्ट 4 to 8 कार्बन एटम के अगर फैटी एसिड्स हैं, सो उसके लिए एक अलग से आइसोफॉर्म है एसाइल कोएंजाइम सिंथेटेज की. अगर इंटरमीडिएट लेंथ है उसके लिए यू हैव डिफरेंट आइसोफॉर्म. एंड इफ दे आर वेरी लॉन्ग चेन फैटी एसिड, देयर इज ए थर्ड आइसोफॉर्म द सेम एंजाइम which activate fatty acids by converting fatty acid into fatty acyl coenzyme A. Activation के बाद these fatty acids they are transported from cytosol to mitochondria, and that is done by a shuttle system called carnitine fatty acyl carnitine shuttle system. And before that, one enzyme called carnitine acyl transferase 1 that transfer fatty acyl group from fatty acyl coenzyme A to carnitine. Carnitine is a modified amino acid which is basically synthesized from lysine and methionine in cell, and this enzyme carnitine acyl transferase ये ट्रांसफ़र करेगा फैटी एसाइल ग्रुप टू कार्निटीन, हाइड्रोक्सिल ग्रुप ऑफ कार्निटीन here, and the product is called fatty acyl carnitine, and ये जो cytosolic coenzyme ये है, this is released here. cytosol में reaction हुई थी activation की, addition of coenzyme A to fatty acid, that is released here, and the fatty acyl carnitine, this is required to transport a fatty acid from cytosol to mitochondrial matrix using carnitine shuttle. So if you look at this reaction again, cytosol के अंदर fatty acid is converted into fatty acyl coenzyme A by the action of acyl coenzyme A synthetase, जहाँ पे ATP use हुई थी, and this fatty acyl coenzyme A, it react with carnitine with the help of enzyme called fatty acyl carnitine acyl transferase 1. So carnitine acyl transferase 1 एक enzyme है which is located in outer mitochondrial membrane. Mitochondria में दो membrane होती है, inner membrane and outer membrane. So in outer membrane which is on the cytosolic side, वहाँ पे you have a membrane embedded enzyme called carnitine acyl transferase 1. So carnitine acyl transferase 1 form carnitine this fatty acyl carnitine from fatty acyl coenzyme A. एक carnitine के साथ react कराएगा and it will become fatty acyl carnitine. अब this fatty acyl carnitine will be transported to the inner mitochondrial membrane, और transport करने के लिए यहाँ पे in outer membrane you have pores, in fact porine structure होती है proteins की जो which work as transporter. So porine के through fatty acyl carnitine will go to the intermembranal space, दोनों membrane का जो space है बीच में वहाँ पे जाएगा transport होके. उसके बाद fatty acyl carnitine will use a shuttle system. This shuttle system is a transporter system which is called fatty acyl carnitine or carnitine shuttle system, जो fatty acyl carnitine को intermembranal space से matrix में transport करेगा. ट्रांसपोर्ट करने के बाद इनर मेम्ब्रेन में एक दूसरा एंजाइम है जो कि पहले कार्निटीन असाइल ट्रांसफरेज 1 का आइसोफॉर्म है, दैट इज कॉल्ड कार्निटीन असाइल ट्रांसफरेज 2, एंड दिस कार्निटीन असाइल ट्रांसफरेज 2 विल कन्वर्ट फैटी असाइल कार्निटीन बैक टू फैटी असाइल कोएंजाइम ए. इट विल यूज ए कोएंजाइम ए जो कि मैट्रिक्स का कोएंजाइम है, माइटोकॉन्ड्रियल मैट्रिक्स का, एंड कार्निटीन विल बी फ्रीड हियर. कार्निटीन फ्री होगा. दिस कार्निटीन विल यूज सेम शटल सिस्टम एंड इट विल गो बैक टू रिएक्ट अगेन to bring another molecule of fatty acid from cytosol to mitochondrial matrix. अब जब fatty acyl coenzyme A मैट्रिक्स में ट्रांसपोर्ट होके आ गया तो उस केस में it will start oxidation. It will go for beta oxidation. The first reaction of beta oxidation will start from fatty acyl coenzyme A. The first reaction of beta oxidation, it is oxidation reaction जहाँ पे fatty acyl coenzyme A which is transported from cytosol to mitochondrial matrix, it is get oxidized by enzyme called acyl coenzyme A dehydrogenase. So this is dehydrogenase enzyme which oxidize beta acyl coenzyme A, और in this oxidation alpha beta desaturation occurs. अल्फा एंड बीटा जो कार्बन एटम है वहाँ से हाइड्रोजन रिमूव होंगे, डिहाइड्रोजनेशन होगा. सो अल्फा बीटा डबल बॉन्ड विल बी इंट्रोड्यूस्ड and same time these hydrogen they will reduce one FAD molecule into FADH2. So in the very first reaction one FADH2 molecule is also produced and the product formed is called trans-enoyl coenzyme A. In fact acyl coenzyme A dehydrogenase जो enzyme है it has 3 isoforms. One isoform for small short chain fatty acid, जो short chain fatty acid है उन पे काम करती है, उनको डिहाइड्रोजनेट करती हैं अराउंड 4 to 8 कार्बन लॉन्ग फैटी एसिड को. दूसरी आइसोफॉर्म है जो मीडियम चेन फैटी एसिड को ऑक्सीडाइज करती हैं, मोर 4 to 14 कार्बन कंटेनिंग फैटी एसिड को MCAD यू कॉल दैट, मीडियम चेन फैटी एसिड असाइल कोएंजाइम डिहाइड्रोजनेज. एक तीसरी आइसोफॉर्म जो वेरी लॉन्ग चेन फैटी एसिड का ऑक्सीडेशन करती है. वेरी लॉन्ग चेन मीन्स more than 12 to 18 carbon atoms, that's called VLCAD, very long chain acyl coenzyme A dehydrogenase. So इस तरीके से there are 3 isoforms of this enzyme which act on different substrate depending on chain length of the fatty acid. The second reaction of beta oxidation is hydration, जहाँ पे जो ट्रांस इनोलाइल कोएंजाइम ए है उसका हाइड्रेशन होगा अल्फा बीटा पोजीशन पे, and the product formed is 3-hydroxyacyl coenzyme. One OH group will be added at beta position, so it will become more activated for oxidation purpose. And this reaction is catalyzed by enoyl coenzyme A hydratase, जहाँ पे एक water molecule use होगा, and the product is basically hydroxy acid, 3-hydroxyacyl coenzyme A. Third reaction is also an oxidation reaction, जहाँ पे जो 3-hydroxyacyl coenzyme A है, it will be again oxidized, and it will be here the hydroxyl group alcohol will be oxidized to have a keto group. So alcohol is converted into keto, and same time one NAD plus molecule is reduced to NADH and H plus. And this step of oxidation is catalyzed by 3-hydroxyacyl coenzyme A dehydrogenase. The last step of beta oxidation is thiolysis, that is catalyzed by beta ketoacyl coenzyme A thiolase, या simply thiolase भी इसको बोल देते हैं इस एंजाइम को, and this convert beta ketoacyl coenzyme A into fatty acyl coenzyme A and acetyl coenzyme A. So इस रिएक्शन में स्प्लिटिंग होगा, लाइसिस होगा अल्फा बीटा कार्बन के बीच में जो बॉन्ड है उसका, और लाइसिस होने के बाद वन coenzyme A is used here. So coenzyme A will be added to the fatty acid, and the terminal 2 carbon atom, these 2 carbon atom, they are released in form of acetyl coenzyme A. So this is the production of acetyl coenzyme A, और जो fatty acyl coenzyme A अभी बनेगा, that will be 2 carbon less. For example, if you started from 16 carbon, first cycle के बाद you will have only 14 carbon fatty acyl coenzyme A here, and this fatty acyl coenzyme A will enter again to the same cycle जब तक सारे carbon atom they are converted into acetyl coenzyme A in beta oxidation. So the number of cycle depend करते हैं कि fatty acid कितना बड़ा है, कितने carbon atom है. If you have 16 carbon in a fatty acid, it means it can give rise to 8 acetyl coenzyme A, and because they are 16 carbon, so it will go through 7 cycle of beta oxidation. लेकिन in case of odd number, कुछ fatty acids हैं जिनमें even mostly fatty acids are even numbered, but some fatty acids are also odd numbers. So in case of oxidation, beta oxidation of odd number fatty acid, जो cycle है that will be repeated as such, except जो final product बनेगा, that final product will not be acetyl. Every cycle will give one acetyl coenzyme A, but when the last 3 carbon will be left, those 3 carbon will be produced in form of propionyl coenzyme A in case of odd numbered fatty acid. And these propionyl coenzyme A, it will be converted into succinyl coenzyme A that will enter into Krebs cycle, that will be used for synthesis of glucose or can be used for production of energy. There is one more thing to remember in beta oxidation. The last 3 steps of beta oxidation, which are catalyzed by enoyl coenzyme A hydratase, beta hydroxyacyl coenzyme A dehydrogenase, and thiolase, the final step. ये तीनों enzyme activity that is present in case of complex form. These proteins form a complex, like in case of, we have seen in case of pyruvate dehydrogenase complex, and in this case this complex is called TFP, trifunctional protein. And this TFP protein, it is basically anchored into inner membrane of mitochondria. Mitochondria की inner membrane में ये तीनों protein complex form में present रहते हैं, and in fact they are found in form of heterooctamer अल्फ़ा 4 बीटा 4 सब यूनिट्स इसकी होती हैं। जो अल्फ़ा सब यूनिट है, अल्फ़ा सब यूनिट में 2 एंजाइम एक्टिविटीज़ होती हैं, इनोइल कोएंजाइम ए हाइड्रेटेज एंड बीटा हाइड्रोक्सी एसाइल कोएंजाइम ए डिहाइड्रोजिनेज। जो बीटा सब यूनिट है, बीटा सब यूनिट कंटेन थायोलेज एक्टिविटी। तो इस तरीके से कॉम्प्लेक्स फॉर्मेशन में जो डिफ्यूजन है सबस्ट्रेट का, चैनलाइजेशन है, दैट बिकम वेरी इजी, लाइक वी हैव सीन इन केस ऑफ पायरुवेट डिहाइड्रोजिनेज कॉम्प्लेक्स। And this 3 protein complex, this is anchored to the inner membrane of mitochondria. And there is one more thing about this. This is only functional. These 3 protein which are formed in TFP, which forms TFP, they only act on long chain fatty acid. अगर long chain fatty acid है, for example you have 14 or more carbon atoms fatty acid, उस केस में TFP उन पे काम करता है। TFP उनको शॉर्ट करेगा और शॉर्ट करने के बाद, शॉर्ट कैसे करेगा? थ्रू बीटा ऑक्सीडेशन। एसिटाइल कोएंजाइम विल बी रिमूव्ड इन ईच साइकिल ऑफ बीटा ऑक्सीडेशन। और जब कार्बन नंबर 12 या उससे कम हो जाएंगे, उस केस में TFP उनको ऑक्सीडाइज नहीं करेगा फर्दर। उनको ऑक्सीडाइज करने के लिए यू हैव आइसोफॉर्म्स ऑफ दीज एंजाइम्स प्रेजेंट इन द मैट्रिक्स इन सोल्यूबल फॉर्म। सो इस तरीके से जो 3 एंजाइम्स हैं, लास्ट 3 एंजाइम्स विच कैटेलाइज लास्ट 3 स्टेप्स ऑफ बीटा ऑक्सीडेशन, उनकी 2 आइसोफॉर्म्स होती हैं। एक आइसोफॉर्म विच फॉर्म ट्राइफंक्शनल प्रोटीन जो मेम्ब्रेन बाउंड कॉम्प्लेक्स हैं, और दूसरी आइसोफॉर्म जो सोल्यूबल फॉर्म में मैट्रिक्स में रहती हैं। सोल्यूबल फॉर्म एक्ट्स ओनली ऑन स्मॉल चेन फैटी एसिड्स। जो TFP है दैट ऑक्सीडाइज लार्ज चेन लॉन्ग चेन फैटी एसिड्स। एंड इफ यू समराइज बीटा ऑक्सीडेशन, so what we got from beta oxidation? In the first step we got one FADH2 and the reactant. In second step one water was used. Third step one NADH and H plus was produced, and in fourth step coenzyme A was used, and finally we got acetyl coenzyme A. And this acetyl coenzyme A, this enters into TCA cycle. It reacts with oxaloacetate to form citric acid and the TCA cycle continues to give energy to the cell. लेकिन जो बचा हुआ है, for example if you started from 16 carbon fatty acid is called palmitic acid, palmitate. So you started with palmitoyl coenzyme A which has 16 carbon total number of carbons, and after first cycle of beta oxidation 2 कार्बन एसिटाइल कोएंजाइम के फॉर्म में रिलीज हो जाएंगे, और जो बचेगा that will be myristoyl coenzyme, myristoyl which has 14 carbon. So this is C14. So C16 is converted into C14 in one cycle. अब this C14 will again enter into beta oxidation cycle, और जब तक all carbon, in fact you have eventually 8 acid because there are 16 carbon. So and acetyl coenzyme may contain 2 carbon of the fatty acid. It means 8 acetyl coenzyme will be eventually produced from complete oxidation of palmitoyl coenzyme or palmitate. So if you look at the reaction, palmitoyl coenzyme, one molecule will use one coenzyme, one FAD, one NAD plus and one water molecule. And what we are getting? We are getting 2 carbon atom shorter, that is myristoyl coenzyme, one molecule of acetyl coenzyme, one FADH2 and one NADH and H plus. That happens in first cycle. But in complete oxidation, to have complete oxidation of C16 palmitoyl coenzyme, you require 7 beta oxidation cycle. So 7 beta oxidation cycle में आपको क्या चाहिए? You need 7 coenzyme. So eventually one palmitoyl coenzyme require 7 coenzyme, 7 FAD, 7 NAD plus and 7 water molecule for complete oxidation. And the products will be 8 acetyl coenzyme, and because they are 7 cycles, last wala, remember the last acetyl coenzyme is produced by thiolase reaction. That is produced directly by the thiolase action by splitting of 2 acetyl coenzyme which are joined together. When fatty acids remain with 4 carbon only, in that case 2 carbons are removed in case of to form one molecule of acetyl coenzyme, and rest of the 2 carbon they form another molecule of acetyl coenzyme. तो इस वजह से in case of palmitic acid, in case of palmitoyl coenzyme, 7 साइकिल होंगे बीटा ऑक्सीडेशन के, but you get 8 acetyl coenzyme A, and then 7 FAD will be converted into 7 FADH2, 7 NADH, NAD plus will convert into 7 NADH and 7 H plus. Right, so this is a complete what you get after complete oxidation, complete beta oxidation of palmitoyl coenzyme A. But we also know that FADH2 that give rise to ATP in electron transport chain. One FADH2 give rise to 1.5 ATP synthesis in electron transport chain, what you call oxidative phosphorylation, and so one NADH that give rise to 2.5 ATP in oxidative phosphorylation. So from here you can further simplify, you can convert FADH2 and NADH2 into ATP. So total ATP from 7 molecule of FADH2 and 7 molecule of NADH and 7H plus will give rise to 28 ATP in oxidative phosphorylation. So these ATP are only from FADH2 and NADH, which are 28 ATP. So you can further write this reaction. One molecule of palmitoyl coenzyme A will require 7 coenzyme A, 7 oxygen atom for oxidative phosphorylation of 7 FADH2 and 7 NADH, 28 phosphate and 28 ADP. And what do you get? 8 acetyl coenzyme A, this is same, it has not oxidized yet. 8 acetyl coenzyme A banenge, 28 ATP and 7 water molecule. That's only from FADH and NADH जो reduced equivalent है उनसे जो ATP बन रही हैं. लेकिन हमें पता है कि जो एसिटाइल कोएंजाइम में है that also enter into citric acid cycle, और एक citric acid cycle से one molecule of coenzyme A that give rise to 3 NADH and H plus, 1 FADH2 and 1 GTP. 1 GTP is converted into ATP. So that can be treated equivalent to ATP. So in one cycle you are getting 3 FADH2, 3 NADH, 1 FADH2 and 1 ATP. And if you convert FADH2 and NADH in form of ATP, considering 1 NADH is giving 1 NADH is giving rise to synthesis of 2.5 ATP, 1 FADH2 is synthesizing 1.5 ATP. तो उस केस में and also consider 1 ATP per cycle produce. So total what you will have 10 ATP produced from one cycle of citric acid when coenzyme A enter into the citric acid. So one coenzyme A will give rise to 10 ATP eventually, and because they are 8 acetyl coenzyme A, so 8 acetyl coenzyme A will use 16 oxygen molecule, 80 phosphate, 80 ADP. So 10 into 8, 80 ATP molecule will be synthesized from 8 acetyl coenzyme A. 8 coenzyme A will be released, 16 carbon dioxide, 16 water molecule. That will happen. Now you can club these two reactions, and what eventually you get from complete oxidation of one molecule of palmitoyl coenzyme A, that is palmitoyl coenzyme A will require 23 oxygenate molecules, 108 phosphate, 108 ADP, and it will release one molecule of coenzyme A. 108 ATP molecule will be synthesized, 16 carbon dioxide will be released, and 23 water molecules. So that is the net reaction what you get after complete oxidation of 16 carbon palmitate into ATP. So you get 108 ATP. So even you can look at the reaction wise also. 7 FADH2 are produced, 7 NADH, 8 then in isocitrate dehydrogenase in TCA cycle. These are the TCA cycle reaction of TCA cycle, and total you are getting 108 ATP. But eventually the net gain of ATP is not 108. In case of beta oxidation it is 2 less, so it is 106 ATP. Why is 2 less? Because remember, in case of activation of fatty acid, when palmitate was converted to palmitoyl coenzyme A, उस केस में 1 ATP was used, and this 1 ATP was not converted into ADP. it was converted into adenosine monophosphate और वहाँ पे 2 phosphodiester bond break हुए थे ATP के. It means that is equal to 2 molecules of ATP. That's why in the net gain you reduce 2 from 108 and your finally what you are getting is 106 ATP from complete oxidation of 1 molecule of palmitate fatty acid. Now the oxidation of unsaturated fatty acids. Unsaturated fatty acids means where you have double bonds, and mostly in case of unsaturated fatty acid you have cis double bond. Cis double bond means the configuration of hydrogen is on the same direction in the double bond. So in that case, if you have cis double bond cannot enter into beta oxidation. So that need to be converted into trans configuration. So one isomerase is required in order to oxidize unsaturated fatty acids, and they do not react at the first oxidation step. They start oxidation from the second step of beta oxidation after isomerizing double bond, cis double bond into trans double bond. That's why in the first step what you get FADH2, that is missing in case of oxidation of unsaturated fatty acids. So per cycle 1 FADH2 will be less in oxidation of double bond containing fatty acids. So for example, if you look at oleoyl coenzyme A, so oleic acid, it has one double bond at 9 position. So how does it get oxidized in beta oxidation? So because up to 9 position they all are single bonds, so it means these atoms up to carbon 9 they will be oxidized using same beta oxidation. 3 cycles will be operated here, and 3 cycles mean there are 6 atoms which are removed from the alpha side, and 3 acetyl coenzyme are liberated like normal beta oxidation process. But after 3 cycle, when this double bond comes here, it cannot be oxidized by enzymes of beta oxidation because this is present in cis configuration. So here you require one enzyme that is called delta 3 delta 2 enoyl coenzyme A isomerase, which convert this double bond, cis double bond, into trans double bond, and this bond is shifted here at beta position, and this product is trans delta 2 dodecanoyl coenzyme A. In this case beta oxidation will start, but in beta oxidation in the first cycle it will enter at the second level, and this with the second enzyme which is hydratase. So it will enter at the hydratase level. One water will act here, so hydroxylation will occur to this bond, and that's why the first step where FADH2 was synthesized in normal condition, that will be missing here. So in this cycle FADH2 will not be produced by this oxidation, and once this cycle is complete, acetyl coenzyme is removed, then further on it will operate in the same way like normal beta oxidation, and all carbon atom they are converted eventually to acetyl coenzyme A. So in case of oxidation of unsaturated fatty acid, additional enzymes in form of isomerase and reductases, they are required to have complete oxidation of unsaturated fatty acids. Now the oxidation of odd number of fatty acids. A long chain odd number fatty acid, they are oxidized in the same way as other even chain fatty acids are oxidized using beta oxidation steps, but in this case the last product is not 2 carbon atom, but there's a 3 carbon atom coenzyme A, that's called propionyl coenzyme A. And these odd number fatty acids, they are present in kettles, in fact in milk, in some plants also. They do, marine plants, marine organism, they do synthesize odd number fatty acids. And in fact in case of जो the propionate है, propionate को mold inhibitor के तौर पे भी यूज किया जाता है in case of cereals and in case of breads, storage के purpose से. So that's how it can enter into the human body propionate. So when propionate is there, either is from food source or by oxidation of odd number of fatty acids, this propionyl coenzyme A is converted into D-methylmalonyl coenzyme A by an enzyme called propionyl coenzyme A carboxylase. So this is a carboxylation reaction, and biotin vitamin H is a cofactor for this propionyl coenzyme A carboxylase, where propionyl coenzyme A is carboxylated and carboxylate होके it forms D-methylmalonyl coenzyme A. And this methylmalonyl coenzyme A is converted into L-isoform by enzyme called methylmalonyl coenzyme A epimerase. So D-methylmalonyl coenzyme A is converted into L-methylmalonyl coenzyme A by epimerase, and this L-methylmalonyl coenzyme A is converted into succinyl coenzyme A by enzyme called methylmalonyl coenzyme A mutase. And this methylmalonyl coenzyme A mutase use B12 cobalamin derivative as coenzyme. So vitamin B12 is used here for this very special reaction जो convert करती है methyl malonyl coenzyme A को succinyl coenzyme A. And we know that succinyl coenzyme A जो है, this is one of the intermediate of TCA cycle. So succinyl coenzyme A यहाँ से TCA cycle में enter कर सकता है, और enter करके it can form oxaloacetate. Oxaloacetate can be converted into glucose. So इस तरीके से जो propionyl coenzyme है, it is also called glucogenic. Glucogenic means it can be converted into glucose, and it enters into citric acid cycle and further can be converted into glucose and other carbohydrates. Otherwise, the another product of beta oxidation acetyl coenzyme A is not glucogenic. That cannot be converted to glucose because vertebrates, they lack the enzyme which are responsible for converting acetyl coenzyme A to glucose. For example, in vertebrates, pyruvate is converted into acetyl coenzyme A. This acetyl coenzyme A can participate into Krebs cycle, can give rise to energy, but this cannot be converted into glucose. Similarly, if acetyl coenzyme is coming from oxidation of fatty acid like in beta oxidation, in that case also this is keto. That's why it's called ketogenic. It is not produced, it cannot be converted into saccharides and sugars. Acetyl coenzyme A is a source of carbohydrate synthesis in germinating seeds. Germinating seeds में जो major source energy का, those are stored lipids. So when germination occurs, energy can be acquired from lipids by oxidation, beta oxidation. Fatty acid converted into acetyl coenzyme A, which enter into TCA cycle, and eventually ATP are synthesized. But when seeds germinate, it requires biosynthesis of other macromolecules also, like protein synthesis, nucleic acid synthesis. So in that case, nucleic acid and proteins, they are synthesized only from glucose. So in case of a germinating seed, the stored fat should be converted into glucose or carbohydrate, and that is done by specialized cell organelle what is called glyoxysomes. So in plants, fatty acid oxidation occurs in leafy tissues in peroxisomes, and in germinating seeds, this beta oxidation that occurs in glyoxysomes. So these plant peroxisomes and glyoxysomes, they have very similar structure and function. Seed में glyoxysome होता है, and जब germinate होके plant बनता है, उस case में peroxisome does that beta oxidation of fats. So glyoxysome occurs only in germinating seeds. So in germinating seed, you have triacyl glycerol जो stored हैं fats. Lipases act करते हैं उन पे. Lipase act करके convert them into fatty acid. Fatty acid का oxidation होगा. Oxidation होके acetyl coenzyme A बनेगा, and this acetyl coenzyme A, it enters into glyoxylate cycle. Glyoxylate cycle में decarboxylation नहीं होता है, और acetyl coenzyme A से eventually oxaloacetate बनता है. and साथ में succinic acid is also formed. So this glyoxylate from glyoxysome it goes to cytosol, and cytosol में जाके it is used for synthesis of glucose by process called gluconeogenesis. अब ग्लूकोज से बाकी सुक्रोज, अमाइनो एसिड्स, nucleotides, other metabolic intermediate, they can be synthesized from glucose, and energy, of course, energy also can be produced from oxidation of glucose. So in case of it occurs only in case of germinating seed, जहाँ पे जो lipids हैं they can be converted into sugars, they can be converted into carbohydrates or glucose. In case of vertebrate, it's not possible to convert fats into carbohydrates. Only in seeds that can happen, and that can happen by using glyoxylate cycle in glyoxysomes. So here in case of germinating seed, more than energy, जो stored lipids हैं, stored fatty acid हैं, उनका seed में use होता है for synthetic reaction, for synthesis of glucose from acetyl coenzyme. And if you compare peroxisome, beta oxidation occurs in both peroxisome and mitochondria. So peroxisome और mitochondria में जो difference है, in that the first difference in peroxisome oxidize very long chain fatty acid. In fact, if carbon number is more than 22, अगर more than 22 carbon है fatty acid में, उसका oxidation होगा peroxisome में. If you have long chain lesser carbon chain fatty acid, they are oxidized in mitochondria. Second difference, peroxisome में जो transport होता है fatty acid का, that is through ABC, ATP binding cassettes transporter होते हैं membrane में, they transport fatty acid to peroxisome. In case of mitochondria, we have seen that जो transport होता है that is through carnitine, acyl carnitine translocase. So carnitine, acyl carnitine जो transporter है वो use होते हैं for transportation from cytosol to mitochondria, whereas you have different type of transporter in peroxisome for the same purpose. Third difference है वो है कि in the first oxidation reaction, in case of mitochondria, acyl coenzyme dehydrogenase ACAD, it produce one FADH2, and this FADH2 enter into electron transport chain and can give rise to 1.5 ATP molecule. Lekin in case of peroxisome, this reaction is catalyzed by acyl coenzyme oxidase, ACOX, and this acyl coenzyme oxidase also produce FADH2, but this FADH2 is oxidized back by presence of oxygen. यहाँ पे ऑक्सीजन एक्सेप्टर की तरह काम करता है peroxisome में, और ऑक्सीजन एक्सेप्ट करता है प्रोटॉन को फ्रॉम FADH2 एंड फॉर्म H2O2 हाइड्रोजन पेरोक्साइड, जो कि इज़ वेरी ऑक्सीडाइजिंग इन नेचर, एंड इमीडिएटली H2O2 बनने के बाद H2O2 is converted into water and oxygen by an enzyme called catalase in the cell, and that's why even the name suggests peroxisome, that is derived from this reaction also, because hydrogen peroxide is formed from oxygen with the help of extracting proton from FADH2. So the environment in peroxisome is very much oxidizing, and this is the third step. So in this case, in case of peroxisome, the 1.5 ATP which was produced in mitochondrial oxidation, that is not there. So 1.5 ATP per cycle का loss होता है in case of beta oxidation in peroxisome. Fourth major difference in beta oxidation in peroxisome and mitochondria is the second and third enzymes, enoyl coenzyme hydratase and 3-hydroxyacyl coenzyme dehydrogenase. These two enzymes present in form of bifunctional protein, whereas in case of mitochondria में these enzymes, there are 3 enzymes in fact. 3 enzymes, they form trifunctional protein. So these are 4 major differences in beta oxidation reactions which takes place in peroxisomes and mitochondria.